Hi everyone. So we'll just wait for maybe like 3-4 minutes till around 6-5 and see if any other people join. Um, can you all see my screen? Just let me know.
Yes, ma'am.
Okay, great. So I think we can get started. As in when people join, I will admit them. Mm, yeah. Okay. So last time we talked about kind of the introduction into this course. We talked about the cell and its structural elements. We talked about the different organelles. We talked about the different um, bonds and interactions that hold together proteins. We also talked about DNA replication, transcription, and translation. So we covered some of those topics um, from your coursework and did some question answers as well. So today we are going to continue with the next week worth of coursework, which has been uploaded. Some of that is going to be about cell cycle regulation and the cell division process itself, mitosis, actually, up till now has been taught to you. And following that, we'll also look a little bit into checkpoints of in the replication cycle, and finally, the ubiquitin proteasome system. So I'll only be talking about a few topics here and there, and what I think would be useful to you going forward. And we'll look at a few question answers as well, where just like last time, you guys can call out the answer once I've read out the question. Um, so I think we can get started now. Right. Okay. Mhm. Okay. Right. So as was covered in your coursework, we know that there are several very essential phases that have been mapped out with years and years of research, uh, dividing this phase into the interphase. Then you have sec. Yeah, you have interphase consisting of your G1, S, and G2. And then you have your mitosis phase or M phase, where you have all of these different phases starting from prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. So I'm sure you're all very well aware of what is happening in each of these phases. So when you start with prophase, most of this time the cell is in interphase. So if you see a resting cell, most of the time you're not going to be able to see all of its chromosomes, right? You're not going to have them condensed. Just a small phase in a cell's life, that is when it actually undergoes mitosis, and that's when it divides. So I just have some text here. Don't be worried about this. You can see it when you are going through these slides later on. But basically, what happens is that in interphase, you have G1, S, and G2. So during G1, you have accumulation of various different nutrients that are required for the cell to be able to synthesize double of everything that's there. So the DNA and certain organelles, you need to make sure there's two of everything, such that when you have daughter cells, you will have equal number and amount of those things from the parent cell. Right, so G1 is basically your growth phase 1, where you have all of these different nutrients that are being accumulated and prepared. And then S phase is your synthesis phase. That's where you duplicate your DNA and duplicate various different organelles that are going to be partitioned into the daughter cells. And that's when the cell is highly metabolically active. And then you have G2 phase, where essentially you have some checkpoints that regulate whether the cell is ready to go into M phase or not. And also the cell increases in size during these phases. So that's interphase, and that's most of the cell's life. And then once it has crossed all of these checkpoints and it's determined to be fit to move forward into M phase, that's when you have G2 transitioning into the first phase of M phase, which is prophase. And here you'll start seeing that the chromatin fibers are condensing into chromosomes that are visible through the light microscope. And you don't need to do anything fancy to view them. Just light or phase contrast microscopy will show you these condensed structures. And these replicated chromosomes, they appear as these two identical sister chromatids that are joined at their centromeres, right? Like this. And the mitotic spindle also begins to form. So these are at opposite poles of the cell. And these will later be what is needed for contractile forces that allow these chromatids to be pulled apart, right? So then the centromeres also start to move to the opposite poles of the cell. That's where your microtubules are going to start lengthening as well. And very slowly, your nuclear membrane starts breaking down. Why? Because the entire cell needs to partition, not just, and so we need to get rid of this nuclear envelope so that everything basically comes into the cytoplasm. So when cytokinesis takes place, everything will be partitioned equally. Then you move to pro-metaphase, the first part of metaphase, right? And here the nuclear envelope completely comes apart. Microtubules will go in and invade this area, the nuclear area. It will bind to the chromosomes. And microtubules bind at kinetochores, which are these disc-like structures that help attach it to the centromere, the microtubules to the centromere. Some other microtubules may bind to each other that allow for contractile forces. Then in metaphase, everyone is arranged in the equatorial plate over here. And this is equidistant from both poles. So now that you're aligned here, and we move on, and there's a checkpoint here as well, which we'll learn about later. We can then move on to anaphase. This is where you have these centromeres dividing and you have sister chromatids that are pulled to opposite ends of the cell. And finally, telophase, where your two daughter nuclei start forming. So you reform this nuclear membrane and start decondensing the chromosomes, and the spindle fibers begin to disappear. And then the cell starts partitioning here at the center. And finally, cytokinesis is when you have two completely separate cells. So your parent cell has now given two identical daughter cells that have all of the different things after being divided at this cleavage furrow, right? So that is the main sort of flow of how the cell cycle proceeds and how mitosis proceeds. And meiosis, of course, is also another process that's there in these germ cells and so on, which will be covered later on in your coursework. And you can find these resources at this useful link I've provided here. So this is a really nice video that I found. On YouTube, but it's from a Zeiss. It's developed by Zeiss Technologies using their microscope. And you will notice that this red, this is marking your DNA or your chromosomes, and the green is some sort of a cytosolic marker. Um, I think it's marking microtubules or, um, and you'll see that the chromosomes are pulling apart. They're aligning, pulling apart, and going to opposite ends, and then you have complete partitioning of the cell. And as your professor also mentioned, that you'll see that while these guys are dividing, they're quite circular, and the ones that are not dividing are nicely spread out. And this is something you see a lot in mammalian cell cultures. I see it all the time where you have, say, a HeLa cell culture or an HK293T cell culture, and you have a lot of cells lying flat. They are not the ones actively dividing, they're all in interphase. These ones that are circular, they are the ones that are dividing. So there are much fewer of them, but you'll see them all around. And some might think that, oh, these cells are not adhering. Actually, it could be that they are actually in division at that very time when you're looking at them. So that's something that you should know for when you do cell culture. And this is another nice image that I found where you have this blue, which is like a DAPI like marker that um, marks your chromosomes, which are condensed at this point because this is metaphase. And you have the kinetochores that are marked with a fluorescent probe, probably an antibody. And they have bound very, very cleanly. So this is doing, this has some sort of reconstruction on the image so that it gets such a clean image. And the green is for the microtubules that are joining at the kinetochores and centromere region of each of the chromosomes. And this, I thought I'd take you through this resource. It's a really nice resource. You can check it out. This link down here on that's showing the stuff. So this is mitosis that's mapped out in these rat kangaroo epithelial kidney cells. So rat kangaroos are actually a marsupial that's found in Australia. And at some point, someone thought it would be a great idea to take their kidney cells and make a cell line out of it. I think it's called PTK2 or something like that. And with this cell line, then they did some extensive fluorescence imaging to look at the different phases of the cell cycle in great detail. So here you have interphase, and this is basically outlining whatever I said earlier while I was explaining. So you have your nuclear membrane here, which is still intact. You have your nucleolus. This is all chromatin, of course. Centrosomes that are still very close to the nucleus, and aster, right? And then all of this is your microtubule network that has not really been organized in any specific way. And they've also marked out the mitochondria that you can see here, right? And so at this point, this is the G2 phase that they have shown. And then the next one is prophase. So after it's crossed the G2M checkpoint, it moves into prophase, right? And here you can see that the chromatin has started to get condensed. These the microtubules have sort of started to localize specifically such that they are going to move to different poles and form these spindle fibers. And the nuclear membrane, although it's not that clear in this image, but the nuclear membrane has started to break down. And the centrosome is also moving away from very strongly perinuclear regions to further edges of the cell. And here you have prometaphase. Like I mentioned, this is where the nuclear membrane is completely disrupted. So like here, you could still see um, some remaining amount of it. And that's, I think there are some porins and stuff that are present in the nuclear membrane that you can mark um, with some antibody or maybe a genetic probe, depending on how you want to carry out your experiment. This is completely absent here. So you don't see any of it. And of course, your chromosomes are a lot more well defined because they've condensed, and your spindle poles have formed very clearly, right? And this is prometaphase, so they haven't really aligned at the equatorial plate just yet. And here now you have later on in metaphase, so you have your equatorial plate here nicely with the chromatids, the chromosomes aligned, and the spindle fibers have all attached to the chromosomes such that they can start being pulled apart in the next phase. And here you have early anaphase. So this is when you start having the kinetochore attached microtubules that they start to shorten and start pulling away. And it's actually pretty fast. And it's actually apparently the shortest stage in mitosis, early anaphase, overall anaphase. And early anaphase will, I think, last maybe just a couple of minutes and maybe depending on the cell type, even seconds. And yeah, so this is early anaphase, so anaphase A, and then there's also anaphase B. I think I have, okay, I don't have an image for that. But basically what happens there is that you have further translocation of the chromatids away from the metaphase plate towards the poles. And so there's a lot of pulling, and you'll read about this probably later, or you can, check it out in some online resources about how it's a very interesting process because how is pulling taking place, right? Who is pulling? Right? That sounds like such a physical or mechanical process, but it's taking place and you're actually pulling this DNA apart. And there's a lot of people who have looked into how this takes place. And you know that microtubules are associated with, these motor proteins. And so you have these these different complexes, Newmark complex, etc., that are accumulated here and actually facilitate the cell being able to contract these microtubules and physically pull apart these chromosomes. So maybe you guys can read about it later if you are interested, right? And so now once everything has been pulled to the separate sides of the cell, and the cell itself would have grown at this point by G2 phase, so that there's enough room in each half of the cell to accommodate all of these different elements like the DNA and the additional organelles and everything. Then they move on to telophase. So this is an image from late telophase. So early telophase would basically have much less of this furrow in the between the cells, and you can see that the chromosomes have started decondensing here. Nucleolus is reappearing because now you're going to start forming a new nucleus in each of these daughter cells. And finally, of course, after this, it moves on into cytokinesis, which is when you have two new daughter cells with all of your decondensed chromosomes. You have a nuclear membrane that's formed. You'll see a nucleolus, which they have not stained in this one. And of course, your microtubule cytoskeleton, which has redistributed to how it typically is, which you have some enriched perinuclear areas, and then it allows for this transport in different regions of the cell. So that is the process, and I think that looking at how it really looks in a cell with fluorescence imaging is very useful for visualizing how it takes place. Um, yeah, and this is something I thought would be very interesting for you guys to know about. So I think it's not been covered, maybe it will be covered later on in your coursework. So it's called a FUSI stain. Okay, so FUSI stands for fluorescent ubiquitination based cell cycle indicator. So it was a probe, it's a DNA based probe that was developed, I think, at least 10, 15 years ago. And it's really, really, at least 10, 15 years ago, yeah. And it really revolutionized the game at the time because with a very simple assay, you'd be able to tell how many generations this particular cell has gone through. So how is this possible? You know that, during the cell cycle, you've studied, right, that different proteins are enriched at different times of the cell cycle. Some proteins are there that help the cell get through S phase, and then they are degraded, with the help of ubiquitination. Other proteins are enriched in G1, some proteins are enriched in M phase. So these CDK cyclins, all of these, right, they are cycling through this cell cycle phase, different cell cycle phases. And so you have, temporally mapped proteins, some that are coming up at one point, some that are coming up another point. And that's what these scientists took advantage of when they developed this particular probe. So it's like a plasmid, right? And it has these different DNA constructs that encode for these fusion proteins. So the first fusion protein is FuCG1 orange. So this is basically a fragment of human CDT1. Uh, that is a protein that essentially is more present in G1 phase, and this is fused to this orange fluorescent protein. That doesn't matter, it's MKO2, but either way, the point is that CDT1, which is enriched in G1 phase, is fused to an orange fluorescent protein, right? And FUC SG2M green, as you can tell from the name, it's some fragment of protein that is enriched during G2 and SG2 and M phases, and that is fused to a green fluorescent protein. Okay, so now what do you think is going to happen? While the cell is moving through the cell cycle, during G1 phase, the cells are going to, in their nuclei, it's going to look orange, right? And while it is in SG2 and M phase, you're going to have a higher green fluorescence in the cell. And in between these phases, it's going to look a little yellow. So using this fluorescence, essentially, you can map out with microscopy or flow cytometry, as long as your cell culture, is starting in a synchronous way, you can actually map out how many generations your cell has, or cell or subset or set of cells has gone through from when you started the experiment, right? Because it will cycle from going from green, and then it'll start with orange, then it'll go to green, in between there'll be yellow, so you can sort of tell, right? Every time it comes back to orange, you're going to have one cycle, so you can count it that way. So this is sort of what it looks like. It's pretty cool, right? You can see the orange to yellow to green transition in many of these cells. I think I can play it again. Yeah. So this was a very, very useful tool that was developed, and actually now people have sort of built up on this um, tool a lot and tried to make different variants of it that are much easier to use. I think making virus versions of this, I think maybe even someone has included another fusion protein, though I'm not sure, that can further label the phases of your cell cycle even better. So you can read about it at these links that I've provided. It's pretty cool. And another thing that I didn't mention earlier on purpose because I thought I'd elaborate more on it here, which is this G0 phase. So now you know that G0 phase is when a cell can exit from your typical cell cycle and it can become quiescent. Not, it can choose to not divide, right? So G0 phase actually can be of different types. It can be of different categories. So I thought I'd go into them a little bit in detail with you. So it can either be of a reversible phenotype, a reversible G0 phase, or an irreversible one. So reversible would obviously indicate that it can easily come out of G0 and go back into the cell cycle and go through the normal phases. So this typically happens, this reversible G0 phase. It's also called quiescence, and it can happen in response to extrinsic signals. That means signals that are coming from the environment. And these cells have been characterized in research in different papers. They have identified these cells to have low RNA content. Why would it have low RNA content? Because essentially you don't need to transcribe more and more DNA to produce mRNA, to produce more proteins, because the cell is not dividing. It's like its metabolic activity is low. So it doesn't need to provide um, the cell with fresh proteins. So turnover is low, low RNA content, right? Then obviously cell proliferation markers. One very well known one is this Ki67 proliferation marker. A lot of people use it in research to mark whether a cell is proliferating or whether it's quiescent, and it's a very common one, Ki67. Yeah. So it will lack these kind of cell proliferation markers as well in quiescence, and increased label retention indicating low cell turnover. So this is kind of pointing to what I said here, which is suppose you have labeled the surface of a cell. Okay. But what happens normally in a cell that is metabolically active is that after some point, if there is a protein on the surface of the cell, it's going to be endocytosed and taken into the cell, and then either it can be targeted for degradation or something else, like it will be chopped up or some used for something. But it will not stay on the membrane surface forever. So that is what is called surface turnover or cell turnover. Okay. So now if you have labeled the cell with something on its surface, and it is and you can actually see with retention time basically how long that label is there, whether turnover is slow or fast. So they are saying that this is increased label retention indicating low cell turnover. Sorry, this should be decreased label increased label retention, that's correct. So that means that the label is retained for longer on the surface because it's not being endocytosed in, because the cell is metabolically slow at the time and there's less turnover. Okay. So an example of this type of reversible or quiescent G0 phase transition happens in tissue stem cells. So stem cells, until they require, until they need to differentiate into some specific cell type, the tissue stem cell is not going to be dividing. So it needs to remain in this quiescent stage. So these cells would have that. Also cells in unfavorable environments, if there's low nutrition, contact inhibition, and lack of growth signals, these can cause the cells to go into quiescence until the environment improves. So low nutrient, suppose in your cell culture you haven't changed the media for a long time. What has happened? Right? It's depleted out all the media. There's not enough FPS. It's not getting enough food. It's not getting enough growth signals. The cells will choose to not divide because it's not energetically favorable. So low nutrient conditions, lack of growth signals can cause your cells to become quiescent. Also, contact inhibition is an interesting one, and this ties in with some cancer cells. I can explain that. So basically, when cells are dividing, if there are too many cells in some specific, fixed area, say this is, suppose this screen is the area and there are cells dividing here. If there are enough cells to form a monolayer where all of the cells are touching each other, and these cells are normal cells, okay, they're not transformed, they're not cancerous in any way, they will have this property of contact inhibition, where if cells are too close to one another, they will decide, okay, let's not divide anymore. It's not favorable because if there are too many cells, we will run out of nutrients, we'll run out of growth signals, and that is not favorable for the survival as a population. What happens in cancer sometimes is that there are some mutations that cause a lack of this contact inhibition. So it doesn't matter key cells are touching each other, the cells are growing on top of one another, they'll just keep growing. And you see this a lot in these cell lines that are derived from a cancer, that if you don't passage them regularly, they will just start growing on top of another. They're not strictly monolayers. They can start forming these globs of cells that you can very easily see. Even without the microscope, you'll see this white blob on your cell culture dish. So that is because of the lack of contact inhibition in these cells and cell lines. So the next type of G naught phase is an irreversible one, which is called senescence. I'm sure you guys have heard of senescence, right? So this is when the cells enter into uh, this G naught phase in response to some DNA damage or degradation. So this can be, some examples of this are telomere shortening. So you know that when cells undergo many replications and they are every time the DNA is replicated and it's divided into daughter cells, you actually have telomere shortening. So telomeres are found at the ends of your chromosomes and they shorten with every single cycle. And so if the telomere has shortened to some extent, actually that leaves the ends of the DNA prone to damage. It's actually dangerous for the cell. That there can be some um, damage at the edges of the cell if the telomere is too short. So that protective element is lost. So if the telomere is too short, essentially the cell is old. If there is ROS production, so ROS are these reactive oxygen species. So if there is some stress to the cell, you have these reactive oxygen species that suddenly come up because of various different reactions within the cell, and they can oxidize and destroy, from lots of stuff, from useful proteins, membranes, organelles. It causes this whole host of dysfunctional things that take place when there's excess ROS. So obviously, if the cell is sensing ROS, and there are many proteins that are involved in ROS sensing, they will shut down the cell cycle and go into this senescence. And of course, oncogene activation is another one. So we know that there are some cells, some genes that are important for cell cycle to progress. So the cell should proliferate. It's pro-proliferation, these oncogenes. And if it is like if there is a gain of function mutation or there is excess oncogene activation for whatever reason, it could be a mutation, it could be some chemical, it could be some genetic manipulation that you are doing for an experiment. In that case, your cell might become senescent because it has detected that oncogene activation has taken place. It's no longer safe to replicate DNA. And if you do replicate it, then that would make the cell's progeny non-viable. So while senescent cells can actually no longer replicate, they will remain able to perform many normal cellular functions. So an example of this is aged cells in renewable tissue. So they're not dividing, but they're performing different functions. Say they are helping with contraction, or they are maybe helping with cell-cell interactions or transport of some nutrients, or they are responding to some stimulus, but they're not dividing. And finally, the last type of G naught phase is this irreversible transition, which is called terminal differentiation. So stem cells that have progressed through a differentiation program, they actually reach a mature or terminally differentiated state. That means that after they have become a certain specific cell type with whatever genetic program that is activated in them, as you know how it happens for stem cells, and they have reached that functional phenotype and maybe gone through some further steps like activation, so on and so forth, they are now terminally differentiated. So they stay in G naught, they perform their main functions, but there's nothing wrong with them. So this is part of, so like how in senescence, essentially the cell has become old or there is some stress. Here, it's just that they have reached the final stage of their maturation and they no longer need to become any further specialized, and they don't provide any progeny either. So some examples of this very well known are myocytes, osteocytes, and neurons. So these cells, basically your muscle cells, your bone cells, and your neurons, initially they come from these progenitor cells that are capable of division, right? They start from like myoblasts, osteoblasts, right? They give rise to cells that eventually differentiate and form these mature cells. And these are nicely performing their function, right? Cardiomyocytes, myocytes, osteocytes, all of them are forming the different structures of the body, and with time they are replaced with fresh cells that are given by the stem cells, but these cells have reached the final differentiation. Right, so with this I thought maybe we can move on to a few questions, right? Does anyone have any questions regarding anything I've taught till now? Just let me know, I can answer. Anyone, any questions? Okay, cool, great. Then I will move on to the questions. Please unmute yourself and tell the answer out loud, as in when I've read out the question. Okay. So, during which phase of the cell cycle does DNA replication occur? Anyone? I hope you are awake.
G2 ma'am.
Hmm, not entirely. Anyone has any other answer?
S-phase. Option
Okay,
B.
that's correct. That's correct. So it's S phase. So S or synthesis phase is when actually the DNA replication, that is the doubling of the DNA content of the cell, takes place, right? By G2, you have already doubled the content and there's a bunch of checkpoints to make sure that it has happened properly. And that's actually also there during S phase. Okay, next question. Which phase of mitosis is characterized by chromosomes aligning in the center of the cell?
Anaphase, ma'am.
Try again.
Metaphase ma'am.
That's correct. Yeah. So metaphase, as I had shown in the earlier fluorescence images, right? In metaphase, you saw that there was an aligning. So in pro-metaphase, it's not aligned yet, but in proper metaphase, all your chromosomes are aligned at the central plate of the cell with the help of all of these spindle fibers and stuff. Anaphase is when actually these fibers start contracting and helping these um, chromatids move apart from one another. So this answer is metaphase. Okay. All right, next one. What structure helps in segregating chromosomes during mitosis? Do you guys know?
Microtubules ma'am. Option B.
Microtubules, ma'am.
That's correct. Well, everyone, that's very correct. So the microtubules are very important. They radiate from the different ends. They join at the centromere with the help of the kinetochores. And then during anaphase, they start separating out into the different, the two opposite ends of the cell. So very good. All right. In which phase do the sister chromatids separate and move to opposite poles? So this now would become very obvious. Hmm.
Option D.
NFH ma'am.
That's correct. Yeah,
Anaphase, ma'am.
that's correct. That's very correct. So to the person who said option D, I mean, you're right in the sense that, in telophase they are separated out, but the movement starts in anaphase, right? So that's why the answer is anaphase. Okay. What phase is usually the longest phase of the cell cycle?
Metaphase, ma'am.
No. Can anyone else try? Not metaphase.
Prophase, ma'am.
G1 phase, ma'am, G1.
That's correct. The person who said G1, you're right. So I mentioned, see here that I did not say what is the longest phase within M phase. Within M phase, then I think prophase is the longest, as far as I'm aware. But within the cell cycle, you have to remember that interphase is most of the cell cycle. And within that, G1 is the longest. So you're right, G1 phase. Okay. This is a true or false. G0 exit can take place from G2 phase of the cell cycle.
Ma'am, false.
Okay.
Ma'am, for
So where does this exit take place from?
S phase.
M phase.
S phase, ma'am.
Mm, so
M
I
phase.
can go back and show you guys the, hold on. What is this here? So it's G1, okay. G0 or G0 typically happens, this exit typically happens from G1, okay. Okay, all right. Cool. So G0 exit, to repeat, it doesn't take place from G2 phase. It actually takes place from the G1 phase of the cell cycle, if at all it takes place, depending on different conditions. Okay, next question. The dash helps in attaching microtubules to the chromosomes at the dash. So can you tell the first dash? So I mentioned it earlier, right? When I was talking about contraction. Sorry?
I thought the first answer is syntosome.
Not centrosome, but you're close. People confuse centrosome and something else a lot. It's not the answer for the first one, but for the second dash, you're close.
Centomere, ma'am.
That's correct. So that's the second dash.
Second, ma'am,
That's
please give me some time. Please,
okay.
ma'am.
Cool.
Microtube,
Everyone think about it. You have time.
chromosome strategy, chromosomes. नहीं मैम सेकंड मुझे समझ में नहीं आ रहा है
कोई बात नहीं. I will tell you. So I
ओके
showed a fluorescence image earlier where there was these pink dots. So do you remember what those were? It was labeled as kinetochore, right? So you will probably study about the kinetochore. You can, read up about it. It's this sort of protein complex. It's a huge protein complex actually, and that is what helps in attaching microtubules to the chromosomes at the centromere on the chromosomes. Okay, so the first dash is kinetochore, and the second one is centromere. So when you look at these images of those, there's very traditional like X-looking um, picture of these two chromatids, the central region, the centromere doesn't have to be right in the middle, but typically you'll see that that is the centromere, and the kinetochore is a complex that helps the spindle to attach to the centromere. Okay, all right, and the last question for this set, true or false? EMS causes chemical mutagenesis as it is a DNA alkylating reagent. What do you guys think? So do you guys remember what EMS is? So I think I'll jog your memory a little bit. So remember when your professor was teaching you in the coursework about the different cell cycle regulating proteins. So essentially, professors at that point knew that cells are replicating, yeast cells are replicating, other cells are replicating, but it was a blank canvas. How is it happening? Are there some proteins that are involved that are regulating one step, allowing one step to happen, stopping the other step from happening. What do you do? So their idea was, and it was generally an accepted method in the field at the time, is that use chemical mutagenesis, right? That means you just randomly mutate a whole bunch of different genes using a chemical agent. In this case, it's EMS, and that can then and then you screen all of these different mutants to see which of the mutants are the ones that have some problem in cell cycle or some change in the way they're performing their cell cycle, and that can let you sort of hone in on what that protein or whatever that gene is that has mutated, how important that is for a particular phase of the cell cycle. So it's a very time consuming and difficult process, but it yields very robust results. So in this case, the answer here is it is true. EMS, okay, which I'll talk about in the next slide. Actually, I have one slide for it. It causes chemical mutagenesis as it is a DNA alkylating reagent. So this is the reagent that they used. Now there are other reagents also that people use. Maybe you guys can read about it. So I'll tell you a little bit about EMS mutagenesis, and I've taken some of this stuff from a very nice, simple YouTube video that you can take a look at. So EMS stands for ethyl methane sulfonate. Okay, so this is a mutagenic chemical. And what happens is that the ethyl group of EMS reacts typically, though I think that it does form some other adducts and products in the DNA, typically reacts with guanine in the DNA, and it forms this abnormal base called the O6 ethyl guanine. So this is what happens. Okay. And then when there is a round of DNA replication, DNA polymerases that are catalyzing this process actually end up putting thymine instead of cytosine across from this weird base, this O6 ethyl guanine. It ends up putting thymine. So now you have this hybrid. So what will happen? The next time there is a DNA replication cycle, for the ones that have this T, you're going to have an A opposited, meaning that in one set of the DNA that has been replicated, you're going to have a complete substitution from what was used to be a GC to a TA or an AT. Okay. So this can occur at, obviously, if you just hit a cell with EMS, you're going to have this kind of a substitution in many different places of the gene. So this can take place in many, many different regions of the genome, leading to this GC to AT transition mutation. And then based on how the clones are looking, how they're behaving with microscopy studies, or these days people can do more extensive fluorescence based studies, you can screen for mutants. Now I think people, because the genome is known to us, and we have all these genome projects that have revealed so much about what different genes, what they function, what their functions are. People have designed CRISPR based screens, right? You guys have heard about CRISPR mechanisms. Basically, it's a way to irreversibly cut out or edit some DNA that is part of, say, some gene sequence or a genome. So you can do this with CRISPR and this Cas9 enzyme, and that is like the new age version of EMS mutagenesis. So you design these probes using these targeting RNAs, and you can essentially just have a screen using these different CRISPR complexes that can go into these cells and form different knockouts, or basically just cut out different genes. And you have a library of these mutants, and then you can screen them in different ways. So that's like a sort of new age version of EMS mutagenesis. And of course, it's much safer because you're not having to deal with this mutagenic chemical that you have to be very careful about, right? Okay, so with that, we are now reaching the DNA damage and replication part. So I'm not going to go into too much detail because there was a lot in your coursework. So you know that in the cell cycle, one way of the cell to detect actual environmental problems is to go into, once it has detected, say, ROS or some lack of food or nutrients or whatever, it can go into G0. But it also has more checkpoints to make sure that transitioning into M phase and moving beyond M phase, you have the correct amount of different raw materials within the cell, and also that the division process has taken place properly as well. So in the G1 phase, this has been taken from your NPTEL coursework itself. So here you can have these different DNA damage stop points or checkpoints that essentially check that there are no like DNA breaks, right? The DNA is undamaged, it's continuous. They can do that with the help of checking whether there's a break altogether in the double-stranded DNA, or if there are some random sort of exposed single-stranded DNA regions as well, because in the cells, single-stranded DNA is not common. Okay, so that is a good indicator that something has gone wrong, right? So in these different ways, you have these DNA damage checkpoints, and you also have a replication checkpoint here, spindle assembly, essentially making sure that the different chromosomes are properly aligned, and it's not like one of them has not been attached to a spindle fiber, or one of them has been attached to too many spindle fibers, so that it does not divide properly. What might happen if your spindle assembly checkpoint is not there due to some mistake or the other? It could be that the pair entirely separates into one daughter cell and it's not divided equally. So that can lead to abnormal cells. So you want to make sure that doesn't happen as well. So you have the spindle assembly checkpoint as well, is a part of your M phase. So these different checkpoints exist. And again, this is from your slides. I just thought I would recap it before going into a little more detail about a few things. So your DNA damage checkpoints here are you can sense this unreplicated or single-stranded DNA. So while the replication process is going on, obviously you melt a little region of the DNA so that you have a template from which you can start synthesizing. But if there was some issue during that process and you have ssDNA or unreplicated single-stranded DNA, this ATR protein is a very big protein that can detect this and phosphorylate CHK or check1, and this phosphorylated check1 will then go ahead and inhibit CDC25, which would have gone ahead to activate CDK2 and CDK1. So basically, it does some inhibitory phosphorylation over here and does not allow the cell to go into the next phase in the cell cycle. Similarly, here, if you have double-stranded breaks or DSB, okay, so both of the strands have some problem in them and they've broken, then it will be caught by this ATM protein. This also is a very large protein, and this can phosphorylate check 2, and that then undergoes the same process. So essentially, both of these two together are detecting these abnormalities and damage in DNA and down-regulating or inhibiting the cell from going further into the cell cycle. And here comes in another interesting protein, P53. P53 is also called the guardian of the genome. And I'll show you an interesting video that I found regarding P53 and how important it is inside regulating cell cycle and making sure that the DNA is, undamaged, and that the progeny cells are not going ahead with some damaged DNA that can cause any host of problems in the person who has the cells. So here one example is that ATM detects a double-stranded break, causes check2 phosphorylation. That leads to the phosphorylation of P53. Now what normally P53 is doing, right, is that it is bound to MDM2 in its unphosphorylated form. And MDM2 will only bind to P53 when it is unphosphorylated. So now what happens when check2 phosphorylates P53? It releases it from MDM2 and it becomes functional, because while it was in the bound state, it was not doing anything. It was just hanging around in the cytoplasm. And Just a sec. Yeah. So, yeah, and when MDM2 is bound to P53, it also causes P53 polyubiquitinalization, and that leads to proteasomal degradation. Okay, and then whatever, and then you recycle, you produce more P53. Again, either it is there nascently in the cytoplasm bound to MDM2, or with subsequent ubiquitination, it can go ahead and get degraded. But the phosphorylated form, that is functional, okay, and that is a transcription factor. So now it will go into the nucleus and it will bind and help the transcription of some specific genes, including P21, which is a CDK inhibitor. Okay, so once the phosphorylated P53 has helped with the transcription of P21 and actually many other genes, these mRNAs will lead to the production of these cell cycle inhibitors. And then this, for instance, this P21, another very well studied protein. This inhibits the CDK2 cyclin E/A complex, and this basically regulates the G1S and SCDK complexes. So these checkpoints are seen at this phase. Okay, so you have these DNA damage checkpoints in the G1S transition, during the S phase itself when the DNA is being replicated. If suppose the DNA was being replicated and the cell was exposed to some sort of harmful irradiation, right? It was irradiated with some gamma rays, and that is done a lot in these um, studies. I know I don't personally do these studies, but I know other students who do. They irradiate cells with some horrific damaging, radiation, gamma radiation and so on, that causes, and they purposely induce these double stranded breaks, and then they sort of chase that double stranded break and see how the cell is recovering from it, or they give some treatment to see if they can improve it, or in a certain cell type that has some mutation, they'll see, oh, but it's not being able to detect this double stranded break. So P53 is very well known, very well studied, and it's known as the guardian of the genome because it is essentially guarding against this kind of a double-stranded break from going on into the daughter cells. Okay, so this is what you need to know about it. And again, a lot of text, apologize for that. But basically, I wanted to talk a little bit about ATM and ATR, right? They're very big protein kinases, and they actually phosphorylate a whole bunch of substrates to achieve their goals. So here you saw that they're phosphorylating check 1 and check 2, but actually that is just one example. They do phosphorylate other proteins as well. So typically, when it comes to ATM, in a normal unstressed cell, ATM is present as a homodimer. Okay, and in that, the kinase domain. So it is a kinase. So that means that it is capable of adding a phosphate to something, its substrate. That's what kinases do, right? With the help of ATP, they add phosphates. So in the unstressed, normal, homeostatic condition, ATM is present as a homodimer, and the kinase domain is physically blocked because it is tightly bound to an internal domain of the protein, that is this serine 1981. That's the position of that serine amino acid. Now, if there is a DNA double-stranded break, so remember that ATM is the one that detects the double-stranded breaks, okay? So if there is a DNA double-stranded break, it can lead to a conformational change in this ATM protein. This will stimulate the kinase to phosphorylate this serine, the one that it was tightly bound close to, it phosphorylates that. And it causes dissociation of the homodimer. What would that mean? That now it is activated and it can phosphorylate different substrates. So it can phosphorylate directly phosphorylate P53, which is nucleoplasmic. It will go into the nucleus, or it can move to sites at the DNA break and then phosphorylate different proteins close to the site of the DNA break itself, like NBS1, BRCA1, which is the breast cancer associated protein 1. So this is very well known to be, mutations in this are well known to be a part of why sometimes some sort of breast cancer can develop, and SMC1. And there are many proteins. These are just some examples. And so then this conformational change that induces this very rapid and extensive intermolecular autophosphorylation. So what does that mean? That it not only is phosphorylating its own self, it's phosphorylating its own serine, it's also can phosphorylate other molecules, right? This does not seem to require the binding of the ATM to the sites of the DNA damage. So it does not have to go and bind at the site where the double-stranded break has happened, but actually it detects it from a little way away. Okay, so that's another completely different mechanism. So there's some higher order chromatin structure which would allow that, and that's kind of beyond the scope over here, so I'm not going into it. So this has also been summarized here. You have an ATM dimer, it becomes an ATM monomer. So this is a homodimer, right? So two of the same thing becomes an ATM monomer. It becomes activated with the help of all of this autophosphorylation. It can then relocalize and phosphorylate these different substrates, leading to cell cycle arrest. Now, ATR kinase. So this is another one, right? Big kinase. And this is actually constitutively ready to phosphorylate. What does that mean? That it does not need to be activated. In the other one, there needed to be some stimulus that's upstream that allows it to become an activated monomer. Here, it is always ready to phosphorylate. But why isn't it always phosphorylating everything then? That is because it depends on the cellular functions that allow the kind of different localization of its substrate. So if the substrate is present somewhere where ATR is not, it will not be able to phosphorylate it, obviously. So regardless of it being able to sort of add phosphates whenever it likes, the substrates need to be localized in the correct place for ATR kinase to go ahead and do that. So this is a little bit in detail. I don't think this is too important for you, but basically it was seen that there was this replication protein A or RPA. Okay, and this one was the one obviously responsible for looking at and detecting these single-stranded DNA portions, right? So this RPA is something that can uh, um, like bind that and stimulate the binding of A trip or ATR to the single-stranded DNA. So it goes and directly binds there, unlike in ATM where it was not directly bound. And this becomes localized to sites where there is this replication fork arrest. So replication fork is formed when you have melted the DNA and you are forming a new strand off of a single-stranded portion, right? But you don't want that to continue if there is some problem at that site. So you want to arrest it at the replication fork. So ATR is found in those regions with the help of this A trip um, binding of A trip to RPA. So this binding takes place and that localizes ATR to these regions of single-stranded DNA. And then any kind of stress like that can cause this. And then this active ATR kinase, once it's over there, then it can phosphorylate critical substrates like RAD17 and CHEK1, and then it can go ahead to sort of figure out what's next and cause cell cycle arrest. And an interesting fact that I read when I was preparing this for you guys is that actually when you completely knock out ATM, so suppose you remove the gene for ATM altogether from the cells, not one of them will have it. Not one copy of the thing is there. Actually, the cells survive. So it could be that ATM is not so essential that the cells will die without it. Maybe there are other mechanisms, other complementary mechanisms that are there that take up the the role of ATM when it is absent. So they manage to compensate for this loss. However, cells and animals that lack ATR, so if you do a whole genome-wide knockout of ATR, so that gene has not one copy in that cell or animal, they actually become non-viable. So it's like embryonically lethal or completely lethal for those cells. That means that ATR's function cannot be done by anyone else. Not any other protein is doing that function. So that I found is quite interesting because while ATM seems to be more, closely regulated, there are other proteins that can sub in for it. ATR, on the other hand, regardless of its constitutive expression, seems to be very, very, very essential. But both of them, of course, are very important for detecting double-stranded and single-stranded DNA, double-stranded breaks and single-stranded DNA. So this is a video that I found. You can access it on YouTube. But I'll show it to you here. I don't know if you'll be able to hear it. If we can't, then you can just follow this link. I'll play it out for you guys. It's quite fun.
This is P53. It's a tumor suppressing protein.
Can you hear that? Just let me know.
Yes, ma'am.
Okay, fantastic. So let's watch it. It's a very nice video and it tells you a lot about P53. Oh no. Okay, hold on a sec.
This is P53. It's a tumor suppressing protein found in cells throughout the body, also known as the guardian of the genome. On a good day, it's all that stands in the way of a dizzying array of cancers. And in the presence of cellular stressors, it jumps into action with the ability to trigger DNA repair, halt cell division, or activate the cellular self-destruct mechanism known as apoptosis. But when it stops working, damaged cells are given the green light to proliferate, with little treatment options to halt their spread. Scientists have been studying P53 for over 50 years. In fact, the gene which codes for the protein, called TP53, is actually the most widely researched gene in the human genome. P53-associated cancers remain difficult to treat. This is because traditional therapies actually rely on P53 to destroy cancerous cells damaged by radio or chemotherapy. Normally, this tumor-suppressing protein is found inactive inside the cell, and this occurs through the binding of a regulatory protein called MDM2. When a cell encounters stressful conditions, MDM2 is released, allowing P53 to be transported into the cell nucleus to save the day. Here, it assembles into a much larger 4-molecule tetramer and binds with specific DNA sequences for gene regulation. The genes which are switched on by P53 are determined by the severity of stress. For example, mild stress may pause cell division for repair, while severe stress may activate cell death. Without the protection of functional P53, damaged cells can spread unchecked, leading to cancerous tumor growth. And this can arise through two mechanisms. The first is through a mutation of the TP53 gene, which leads to a faulty misfolded protein. The other instance is through an overabundance of MDM2, deactivating too much P53 in the cell. Although many are under development, there are still no effective catch-all treatments for P53-associated cancers. Promising strategies involve restoring activity of mutant P53 or reactivating P53 that is bound up by too much MDM2. There are a number of drug candidates under investigation to tackle both of these strategies, but none of these are likely to solve the entire problem. Instead, combining P53 restoration or reactivation with traditional cancer treatments is likely to pack the most powerful punch. So-called combination therapy would involve a cancer-fighting super duo, providing functional P53 in cancer cells and then hitting them with chemo or immunotherapy, destroying the cancer cells once and for all.
Right. So that was a very dramatic representation, but it is quite accurate in how P53 is important. And of course, aside from P53, there are many other tumor suppressor genes that are involved in making sure that there is no unchecked sort of damaged DNA that's going forward into daughter cells, but it's not infallible. So there are mutations that can cause P53 to become non-functional, or the regulatory element of P53, that is this MDM2, it can cause excess copies of that. So you just have P53 being sequestered and ubiquitinated and sent for destruction without it being able to do its job, right? So here I have a little bit about this TP53. It's apparent, it seems to be the most frequently mutated gene in human cancers. So it is detectable in about 50% of human breast cancer, colon, liver, prostate. I mean, it's not important for you to know which cancer specifically, but across all these different cancers, 50% of them will have this TP53 gene that's mutated in some way or the other. So normally, as I had said, wild type P53, when if there is some DNA damage, it would act in a restraining way. It would make sure that cell replication does not take place until the damage is repaired. And if the damage cannot be repaired, then the cell is essentially decides to go into apoptosis. That means that cell is destroyed. That damage is beyond repair, so that cell does not need to then proliferate further. So it goes into apoptosis. So that also p53 as well as many other genes are very important for. So it prevents the propagation of these DNA defective cells and it doesn't lead to cancer phenotype. But if these mutations are there, it can affect the cell cycle. They lose control of cell proliferation. It leads to propagation of damaged DNA into the progeny cells, which become transformed into cancerous cells. And when if you go into biology and you guys are working with cell lines, you'll have these different cancer cell lines. And something that we have seen is that you can have different varieties of cancer cell lines, some that have a p53 knockout or a p53 mutation and some that don't. And something that we have seen in our lab is we are working on a specific type of a specific type of cancer cell line. We saw that the ones, when we implant them for tumors, we saw that the ones that have p53 mutations actually are growing faster and they're causing a much more severe disease. So when we're trying to, develop a cure or we're trying to test some sort of therapy, actually, we see that the one that doesn't have p53 is a much more sort of dangerous and, very proliferative kind of cancer. And that's seen in other different types of cell lines, like for different types of cancers. And this is a little sort of graph set that I took from this paper, where you can see that for patients, right, who had breast cancer, so these are the number of patients, right, or pancreatic cancer or hepatobiliary cancer, we see that in unaltered group, that is basically healthy people. Okay, so people who don't have, sorry, I mean, these are patients who don't have the p53 mutation in their particular type of breast cancer. So they have breast cancer, but they don't have the mutation. Whereas red one or altered group is the one where they have some sort of mutation. So you'd see that this is not, of course, the case always with every single cancer, but you can see that in this type of breast cancer, if you have a p53 mutation, actually the survival over time is lower. Overall survival is lower, right? This also here you can see in the case of pancreatic cancer that you have uh, overall this sort of lowers down until people just don't survive beyond 24 to 28 months. That's when it really plummets. Whereas others, they might even have a very nice long progression free survival or PFS. But then again, in hepatobiliary cancers, it's not really the case. It's pretty similar thing, and it seems that for at least a long time, the trajectory is similar until a point where you see that this effect is really showing up phenotypically in the patients. And of course, there is probability in this data because people would have undergone different kinds of therapies, but I think they would have categorized the patients based on at least a similar type of therapy that they've undergone. So anyway, the point is that P53 is very important for guarding the genome and making sure that defective DNA doesn't progress. And cancers can come up when there's this unregulated growth because of the mutations in P53, and that can make them very potent. Okay. So that is something I thought I'd want to impress upon you guys before moving to the questions. So now from whatever we have learned till now, what do you guys think? P53 is which of the following? Do give me your answers.
Tumor suppressor gene.
That is correct. It is also some other things. Do you see any other option that matches P53 in its description?
Option B में.
That's correct. So it's B and C. Do you
Option
guys all
D.
understand? Yeah, correct.
Option C.
Yes, it is
Transcription
also C.
section.
So it is a tumor suppressor gene. Why is it called tumor suppressor gene? Because of what I explained, right? That it's making sure that the cell does not proliferate too much or does not proliferate in an uncontrollable way. So if it can do that, it is suppressing a tumor. So tumor suppressor gene. but it is also a transcription factor. Like I said, once it has been released from MDM2, phosphorylated p53 translocates into the nucleus and helps as a transcription factor with the transcription of various important genes that are involved in cell cycle regulation. One of them is P21, right? So both B and C is the answer. So here it is D. Okay. Did everyone understand? Everyone's getting it, no, till now? I will move on to
Yes,
the
ma'am.
next one. Okay,
Yes, ma'am.
great. Cool. Now, true or false? CDKs are only activated by phosphorylation. Or actually, I should have phrased it better. CDKs are activated by phosphorylation only. Yeah,
False, ma'am.
that is correct. Does anyone want to explain why it's false? Anyone want to try and take a stab at it? Anyone? So they're activated by phosphorylation. That's very correct. But phosphorylation can also do something else to CDKs, right? If you guys remember, right? It can also inhibit CDKs. So how is it happening that phosphorylation can do both of these things? Then why isn't it being activated and inhibited at the same time? So does anyone know why? No? Think about where phosphorylation occurs, right? It is not occurring, you know, say on just some random spot on the protein. There are some specific sites, right? So there are some specific amino acids that are typically phosphorylated. So in a CDK, in that protein, if you have some, uh, if you have phosphorylation in some specific residue, that would cause conformational changes that cause it to be activated. However, if you have phosphorylation in some different residue, okay, in some other area of the protein, that causes different conformational changes, or it blocks some site or does something else, then in that case, it can be inhibited. So basically, phosphorylation can do both of these things to CDKs, both activation and inhibition. Just depending on where you are phosphorylating in the protein, that is going to dictate which of these two it will happen. Okay. So the answer here is it is false. It's not just activating with the help of phosphorylation. It can also inhibit based on where. Okay. So yeah. Okay, this is another one. So you guys remember V1, right? In yeast, that was something that was part of the coursework, uh, right? So what phenotype would the daughter cells have if you were to say lose this V1? If you were to say genetically delete V1 in yeast or mutate it so that it's not functional anymore, right? And it's not part of your cell cycle regulation cascade, then what would the daughter cells be like? What would they look like? Any idea? Anyone?
Option C, ma'am.
Option C. So not entirely. I think someone sent a message saying option A also.
Larger cell.
Someone is saying larger cell. Can you guys think a little bit more about the V1 function? Okay,
Smaller cells.
that's correct. Yeah. So yeah, it's not larger cells or cells with so many X genetic material. Actually, it is smaller cells. The daughter cells would be smaller because V1 actually decides when mitotic entry will take place, and typically loss would results in premature entry into M phase. So what would happen then? The cells aren't big enough yet. They're not ready. They don't have enough area to accommodate. So actually the cells are smaller. If you lose V1, that cannot that regulatory element is for the transition is lost, then actually the cells turn out to look smaller. These are yeast cells. Okay. So yeah, basically it inhibits the entry into mitosis through inhibiting CDK1 in yeast. It has other homologs also in mammals and stuff, but in yeast it is called V1. And it inhibits CDK1. Oops, and I gave the answer here. So V1 inhibits CDK1, which I've given you the answer of, by phosphorylating it on two different sites, dash and dash. So it was there explained to you in the coursework. Do you guys remember by any chance what sites? This is just a memory question, really, but it's nice to know that it requires two sites to inhibit CDK1 before the M phase transition. So do you guys have any idea? You don't have to say the exact amino acid position. Maybe you can mention what is the amino acids involved that typically are phosphorylated, right? So can you phosphorylate a valine? Can you phosphorylate like a, I don't know, a glycine? Is it any of those? Or are there some specific amino acids that are more amenable to being phosphorylated?
Tyrosine and tryptophan.
That's good. Yeah, and there's one more. Threonines can also be phosphorylated. So in this case, but then really good, you got two of them. So the correct answers here are tyrosine 15 and threonine 14. So those are the two sites that are phosphorylated in CDK1. So yeah, that is a question. If you guys are like tired and want to break or something, let me know. Otherwise, I'll keep going. Do you guys want to break? Are you good? I guess you're good. Then I'll keep going. All right. Okay. So this is about the spindle assembly checkpoint or SAC. This is active in which phase of cell cycle? Think about it.
M phase, ma'am.
That's correct. Does anyone have any other answers that they're not satisfied with this one? They think it's something else? Everyone is satisfied? So yeah, it's correct. It is M phase. And specifically, in fact, spindle assembly checkpoint is most active between um, I think between pro-metaphase till anaphase. Like by anaphase, it will basically determine whether the cell goes into anaphase or not. So from pro-metaphase onwards, up till just before anaphase is when your spindle assembly checkpoint is active. So it's making sure that all of the cells, all of the chromosomes are binding correctly and in the correct number. So not too many of them are binding to one thing. One is sending a message. No, I don't know. And it makes sure that the chromosomes are poised to be correctly divided into the daughter cells at anaphase. All right. So an effector in SAC or spindle assembly checkpoint is the MCC. Okay. So do you guys remember from your coursework what MCC stands for? So it's a okay. I'll let you guys think. No?
Ma'am,
Yeah.
the second question is APC dash which is required remove sister chromatid cohesin. That is what mean? मतलब यह कि एक सिस्टम को जो होता है उस प्रोसेस का नाम बोलना पड़ेगा ना?
अभी फिलहाल तो फर्स्ट का ही बताओ कि एमसीसी का फुल फॉर्म क्या है, देन आई विल कम टू दैट एंड एक्सप्लेन द क्वेश्चन और अच्छे
Okay,
से,
ma'am.
है ना? तो the first one I'll just tell you guys since I think maybe this is a little bit confusing. So MCC stands for mitotic checkpoint complex. Okay, so for spindle assembly checkpoint to take place, for this entire process to happen, you need to have this mitotic checkpoint complex. Okay, so that's just a bunch of different proteins that assemble and allow this checkpoint to proceed and inhibit um, different molecules involved with moving forward with the cell cycle. And I'll talk about it, I think, in a little more detail in a slide after this. So now coming to the next question. So this complex, this MCC, this inhibits the activity of the APC. So what is APC? Do you guys remember? Then I will also mention what I mean by this dash.
APC means, ma'am, antigen presenting cell.
No, not in, okay, it is antigen presenting cell. So, and here, sorry, I meant this to be of the APC and then in brackets, I'd say stands for. So, APC in this case is not antigen presenting cell, that's in an immunology context. But in a cell biology, cell cycle context, do you think you know what APC could stand for? It's another complex. So, the C is complex. But what is A and P? So I mentioned, right, that spindle assembly checkpoint, which is in M phase, right? It is active from pro-metaphase up till just before anaphase, right? Meaning that it is, if it were to detect that there is some abnormality in how these chromatids are going to be separated, it would stop anaphase from happening. So that means it would have to interact with proteins that are important for the functioning of anaphase, right? For anaphase to progress. So APC, therefore, in this context is anaphase promoting complex. Okay. So there's a bunch of different proteins that are involved that help to remove sister chromatid cohesion. What does that mean? So the sister chromatids are joined to each other, right? So there is an anaphase promoting complex that helps make sure that these are not stuck to each other and they are actually separated from each other while they're being pulled to the different ends of the cell. So that is this. There was a typo here. That's my bad. This APC ke baad it should have been stands for. Anyway. Yeah, so that is about spindle assembly checkpoint and the mitotic checkpoint complex and the anaphase promoting complex or EPC. So you guys can read about this and how they kind of this whole process takes place and how it makes sure that the final step of separation is not happening. Another if there is some problem and something interesting you will think about, right, is that if it doesn't happen, what will the cell look like, right? It's going to be this weird cell that has double the amount of DNA that's sort of frozen in time until the cell figures out what to do next. So this is actually something also you see in these cultures that have these cancer cells. You see it a lot. You'll see these cells that are fused together and they're not really going forward, they're not going backward. You have cells that are diploid. So you have, sorry, diploid, quadruploid, they have too much DNA because there's some mutation that is affecting their checkpoints. And so somehow, there is incorrect division of the genetic material. So one daughter cell ends up having, double the amount of DNA that it should have. And so you will see that in these kind of cancer cell cultures where you have, diploid, triploid, quadruploid, all sorts of random things happening just because the cell is no longer able to regulate at the spindle assembly checkpoint. So that's a real world example, and it happens a lot in these cancer cells, in humans, genuinely in their tumors, as well as in culture as well. And so this is in more detail. As I said, I would just go into in brief here. So this MCC that I mentioned, right, the mitotic checkpoint complex, this is triggered by unattached kinetochores, meaning that if it is not attached evenly, this one is just going to go into this daughter cell. So this daughter cell will have double the amount of this particular set of chromosomes. Okay. So it's triggered by that. I'm guessing this, I don't know myself actually, but I'm guessing the reason it gets triggered is that there must be some exposed proteins and stuff here that cause a cascade that eventually triggers this. And this leads to this MCC ka formation. So you have these different proteins that form this complex. Your professor talked about the MAD proteins, right? So here you have MAD2, you have CDC20, BUB-R1, BUB3. So she mentioned the BUBs as well, right? And this MCC, this inhibits APC, right? So the anaphase promoting complex. And this APC is actually an E3 ubiquitin ligase. So what does it do? It targets securin and cyclin for degradation. And by degrading securin, it releases separase, whereas cyclin B, if you degrade, it leads to CDK1 inactivation. And these degradation events are what cause the chromosome segregation and mitotic exit. Basically, you need these degradation events to happen so that the cell can go into the exit mitosis and basically finish mitosis, okay, and form the daughter cells. So in pro-metaphase, the mitotic checkpoint signaling is triggered by the presence of unattached kinetochores, as I showed you here, leading to the assembly of MCC with these different proteins. And this complex then blocks the ubiquitination activity of APC as such, and this does not allow it to then because it's a E3 ubiquitin ligase, and we'll talk a little more in detail about the ubiquitin proteasome system in the next couple of slides. So basically, since it's not able to finally attach ubiquitin molecules onto its target, cyclin B1 and and securin, that does not allow the cell to then move on and perform chromosome segregation and mitotic exit. So that's how it happens. So to anyone who was confused earlier by the questions, I hope this is sort of explaining what's taking place with the help of these different complexes in cells. Does anyone have any confusions or questions? I can go into that. All good? Okay, then I will move on to the next thing. Oh, okay. So right away, I go into the ubiquitin proteasome system. So right now I was talking about it. So I thought I would just recap what you guys learned in the lecture. And then we can talk about some question answers after this. Okay. So the ubiquitin modification essentially is needed on different protein substrates, and that can lead to different types of um, basically target the cell for different functions. Okay. So this happens. How do you add the ubiquitins onto some protein? So you start with E1. Okay, so E1 here, and this is free floating ubiquitin. So what happens is, with the help of ATP, okay, so energy dependent, it has ubiquitin added onto it. So ubiquitin is a very highly conserved 76 amino acid protein, and this reaction allows E1 to bind to ubiquitin, one moiety of ubiquitin with a thioester bond. And then this reaction allows the further binding of ubiquitin to E2. So essentially you're swapping out E1 and putting E2 there. And after this, we bring in the protein substrate, where with the help of E3 and the protein substrate and E2 in the process is swapped out to have, with the help of E3, essentially it's sort of like catalytically forming an isopeptide bond between the carboxy terminus of ubiquitin and the lysine residue. So ubiquitins are added onto lysines of the substrate protein, and this is with the help of this ubiquitin ligase enzyme. So while E2 is swapping out, essentially you have it holding on to the protein substrate, and then E3 allows the ubiquitin to move on to the lysine residue on the protein substrate. So now you have, okay, now you have a monoubiquitinated substrate, protein substrate. And then this can undergo multiple cycles and you can have different amounts of ubiquitination. And this ubiquitination or multi-mono ubiquitination can target these proteins for endocytosis, DNA repair, protein localization, and trafficking. So it kind of depends on um, the substrate, like a certain subset of the substrate proteins. So the E3 ligases help provide this specificity. So you have many different E3 ligases, not just one in the cell, and they function based on so each of them will modify only a subset of these substrate proteins. And also further on to further improve specificity, also post-translational modifications. So guys know what PTMs are, right? So it's basically different groups like phosphorylation, glycosylation, sumoylation, blah, blah, blah. All of these different PTMs are added on to proteins that functionalize them in different ways and also help with substrate specificity when it comes to the final E3 ligase based ubiquitination step. So based on what needs to be done to the substrate and what is the PTM sort of PTM functionalization of that protein, the E3 ligase that is required will be recruited, and that is the one that will start with adding on these ubiquitin molecules to specific locations on that substrate. And that will determine what is going to happen downstream, what is going to happen to that protein substrate. Now, also you can have from monoubiquitinated state, you can also have polyubiquitination. Uh, and based on sort of where you have these ubiquitin molecules, you can have different structures of ubiquitins. Like you can have chains, you can have these sort of globular formations, and that can target these protein substrates based on how and where they have been ubiquitinated for different functions. So they can be targeted because there is some DNA damage response, there is some weird protein that's forming maybe. So this substrate will then be sent for degradation. Or maybe it's maintaining the mitochondria, making sure that mitochondrial DNA is intact. If there is some problem, it can lead to mitophagy. Lysosomal degradation, if this protein is, say, some unfolded protein, it's some abnormal protein, some dangerous or defective protein, it can be targeted to the lysosome so that it can digest it. TCR signaling, that's in regarding the T cell receptor. And of course, it can also activate NF-kappa B signaling. That is basically is activating inflammatory pathways in the cell. And this type of this type of ubiquitination can also lead to 26S proteasome recruitment, which essentially is this barrel-like structure that takes this ubiquitinated substrate into its barrel, and then in an energy dependent process it breaks it down, and then these peptides are then recycled and used by the cell. So it's very efficient, and based on this kind of thing that I'll go into, you can actually, oh, someone is saying they're not able to see the PowerPoint. Oh no, can everyone see the PowerPoint? Are people having a problem? Can you guys see the PowerPoint? Oh,
Yes, ma'am.
oh, you can see it. Okay, great, great. Okay. All right. To the person who can't see it, I am sharing it such that everyone can see it. Maybe, I don't know, try like exiting the meeting and coming back again. Yeah, my voice is heard. Okay. Uh, Maybe try exiting and coming back. That might help because I think everyone else can see the PowerPoint. Yeah. Let me know if the problem persists, but I don't think I can really do anything from my side. All right, so that is about the basic sort of flow of what is happening with the ubiquitin proteasome system. And I talked about E1, E2, and E3. which are these different activating, conjugating, and the ligating enzymes involved in making sure that a protein substrate has ubiquitin molecules attached to it, and then it's targeted for different functions within the cell. And what is, interesting to know is that ubiquitin actually has 7 lysines. So I mentioned that these molecules are attached onto lysines, right? So polyubiquitinylation, I think I spelled it wrong here, can generate linear or branched chains with different topologies. So you have here ubiquitinated, oh, this is hard to say, substrate, and that can lead, that can be a part of DNA repair, it can be part of viral budding, gene expression, endocytosis, all these kind of homeostatic functions. If you have a substrate that has single ubiquitins that are attached to it, but at different regions, so it is mono-ubiquitinated, that can target it to endocytosis. So it's suppose on the surface, then it will be endocytosed within the cell. You also have branched ubiquitins that are sort of all attached to one another and starting from one that is attached to the substrate. Then these are attached to each other via different lysines that can modulate its activity. I guess that's one example. And then you have these ones that are fully polyubiquitinated, and that can lead to proteasomal degradation, DNA damage, tolerance, signal transduction, all these different functions, NF-kappa B activation. So based on where and how, as I mentioned, poly and mono ubiquitination can uh, target the substrate for different types of functions or degradation and try to save the cell sometimes from unfolded proteins and so on and so forth. So I have some questions left here which we can discuss. I think this is the last set of questions. So we'll go through them one at a time. Which of the following is true? What do you guys think? I hope everyone's awake.
Option B, ma'am.
Option B. B for ball?
Yes, ma'am. Yes.
Okay, someone has messaged C, you are saying B.
Option D.
Yes, I think the person now, you got it right now. So it is D, D for dog, all of the above. Why? So because B, C, they're all correct, but A, you guys are skipping out on, right? So I mentioned that ubiquitination occurs on lysines. So if for those who remember that the single letter code for the amino acid lysine is K. It's weird and it's difficult to remember, but you saw here, right, that this K11, K48, K63, K this, K that. What does that mean? So those are lysines. And where and how those lysines are linked, that is giving this kind of structure of these different ubiquitin molecules stuck to each other. So those were all lysines. And because one ubiquitin has 7 lysines, that's why you have such a diverse way in which they can attach and form linkages with one another, right? So in this one, all of them are correct. It occurs on the lysine. It indeed occurs in the cytoplasm. And the final degradation, of course, takes place within the proteasome, like in the 26S proteasome. So all of the above. Right. So next question. Ubiquitin is covalently attached to the target proteins via what kind of bond?
Option B.
Option B. Okay. Does anyone have any other answers? Anyone want to contest? I think everyone is tired now, so I'll just get through it. So it is correct. You're right. It is option B, isopeptide bonds. That's how ubiquitin is covalently attached to the target proteins or the substrate. So now you might now wonder what is an isopeptide bond, right? You've heard of peptide bonds, right? When you exclude an H2O and you bind a COOH to an NH2, and you see it in a lot of these amino acids binding to each other to form these polypeptide chains, right? In the um, to form proteins. So what is an isopeptide bond? What is going on there? So typically when you see iso, you remember that, okay, so this is on another side. So that's how I remember it. An isopeptide bond is also a similar amide bond only, but here it forms between a COH or NH2 of a side chain. So there, when it came to amino acids, which have the peptide bond in between them, it is of the alpha amino group and the alpha carboxyl group, right? So if you remember the amino acid structure, right, you have a side chain, you have the top, which is you have the H, right? One side is an H, and then you have on one hand a COH group and one hand an NH2 group. And side chains can also have COH or NH2 groups, and that can determine, their basicity, acidity, all of that, charge, and all that. So what happens in a peptide bond is the alpha group, that is the one that's bound to the main that carbon. Those are the ones involved in the peptide bond. But isopeptide bond is when the NH2 or COH in the side chain is what's involved in forming this type of peptide bond. So it's a type of peptide bond, and this is the one. And that would make sense, right? Because if you look at these images, and you're forming branch structures, you would not always just form linearly like this one. For instance, there's alpha amino linkage that's involved. This would not always be the case. So that's why you have to also sometimes, based on if you want to have branching or not of the ubiquitinated structure, you would have to then also form this isopeptide bond with a side chain. Cool. So, all right, this is the next question. Okay. Which family of ubiquitin E3 ligases acts as an adapter and does not form a covalent bond with ubiquitin? This is just from the text that and the videos that your professor has uploaded. So I thought I'd just see if you guys remember. Does anyone remember? And maybe you can tell me what this particular family is if you want to. Hmm.
Option D, ma'am.
Option D. No. So actually the correct one here is the ring family. So I also am not that familiar, to be honest, with this, but basically what I have read is that ring is a really interesting new gene. So that's a fun name, family of proteins, and it's a type of E3 ubiquitin ligase, and it functions as an adapter. Uh, and it does not form a covalent intermediate with ubiquitin. So it's basically um, it binds both the E2 enzyme and the target protein via the ring domain, and it holds them closer together. So it promotes direct ubiquitin transfer. So it does not need to have, covalent binding. It sort of just brings them in the vicinity of one another, and this allows it to happen quite quickly. And the ring family has hundreds of these different E3 ligases. It's the largest class of E3 ligases in mammals. There is about 600 of them in humans. And yeah, so that's what they do. So they don't form that, that thioester intermediate that I talked about, right? That thioester intermediate is not formed here. They just directly, they like sort of temporarily hold it, and then they transfer it from E2 to the substrate with the help of just bringing them close to each other. So that's the ring family here. So that's option A. That's the answer. All right, an easy one for you. True or false? Does monoubiquitination target proteins for degradation or not? What do you guys think?
True, ma'am.
Mm,
D.
so not entirely. Actually, typically polyubiquitination is what targets a protein for degradation. Yeah. So that's just how it is, I guess, in the cells. That's, I guess, the polyubiquitinated like chains. Those are how kind of detected. Um, and that is what summons the 26S proteasomes so that it can be degraded. Okay, and I think this is the last question. So this is an open question I thought we could discuss. So I showed you the pathway in which different substrates are ubiquitinated, right? And it involved E1, E2, and E3. So why do we need E2? Right? Why don't we just directly jump from E1 to E3? What is the point of having E2? It's just transferring ubiquitin, right? So why do we need it there? So I'll take you back to this thing so you can take a look at it. And if anyone has an idea, or you just think about it, there's no right or wrong here really. Why is E2 there? E1 is nicely sitting with ubiquitin. Okay, then E2 grabs it. And then E2 transfers it via E3 to the protein substrate. But this could have just as well been E1, right? This whole step is there. But this E1 could have held the ubiquitin, and then E3, with the help of the ligase, it could have transferred to protein substrate. Why is E2 there at all? Is it redundant, or is it some genuinely important part of the ubiquitination cascade? What do you guys think? Does anyone have any sort of guesses as to why E2 is there at all? Why have we evolved to have E2 at all as a part of this cycle? Any ideas? You can guess. I mean, really, there's not too much right or wrong here. If anyone has any clue, what could be the possible importance of E2 here? Because just from face value, it just looks like it's an extra step that the cell is going through, just making all this E2 just so it can just transfer to the substrate, which E1 could have done. Why is it there? So I think time is of the essence now. So let's just um, cut to the chase. So actually, there is a paper that I have linked in the last slide, okay, where I have asked this question, right? That's very nice paper. It's interesting and it talks about the importance of E2. It's not just some adapter. It's not just this random step that's there and people don't know why. There's a paper that talks about the importance of E2 as a part of the ubiquitin proteasome system, but some of the points from that, someone is joining now. So one is the specificity in ubiquitination. So what happens is that there's not just one E2 enzyme. Just like there's not one E3 ligase, there's also different E2 enzymes. And they work with specific E3 ligases. So that makes sure that the type of ubiquitin modification, monoubiquitination, polyubiquitination, which lysine it's transferring to, this fine tuning will happen a lot better than if E1 were to transfer directly. There are many E2s that correspond to different E3s and different modifications. So it fine tunes that specificity. It's also, it's important as an adapter molecule. So having an extra step apparently would increase the fidelity of the transfer. And also different E2 enzymes contribute to the formation of specific ubiquitin chain linkages. And of course, that could then determine the fate of that protein. Degradation, some signaling function. So E1 alone does not have this flexibility. E2 is the one because it has many different types. It can determine and contribute to making sure that it's the right kind of modification. Also, there is energy conservation. So E1 uses ATP, right, to activate ubiquitin, and it does not actually remain bound to it for too long. So by passing ubiquitin to E2, you actually become more energy efficient because you allow multiple E2s to interact with a single E1. So you can sort of branch out the process that way. Yeah. So, and one, I think, last point that I had read about was that E1 enzymes actually have a very conserved function. So there are fewer of them and they function in one particular way, which is ATP dependent ubiquitin uh, activation, ubiquitin binding, right, via thioester linkages. But E2 and E3 actually have more adaptability and they have many different types of E2 and E3. So that basically points to the earlier points that you have um, basically different permutations and combinations that you can have of E2 and E3 that can determine how your substrate is going to be dealt with in the cell, what's going to happen to it. Yeah. So that is sort of the importance of E2, because sometimes you look at these different processes and you think, oh, this is so, well charted out. But then you look at one step and you think, okay, but the cell could have gone ahead without it. But it turns out that there is a reason for everything. And these are few of the reasons for having E2 around. So that's it. That's it for today. I think I have covered everything. Yeah. And I will upload this PPT as well to the Google link thing that shared with you guys, and all the references I have taken stuff from. For that E2 paper, this is the reference I was talking about. If you guys want to check it out, you can take the link from there. Yeah, if anyone has any questions, please let me know. I can explain things. Uh, yeah, everyone's all good. Can you send the Google link? Uh, sure. You guys have it, I think. If you look in the coursework, like your portal, right, for this course, they have shared the link for one Google Excel, like a what is it, a Google Sheet, and that has links to the YouTube videos where essentially I'm uploading the recording of this this session, as well as the link to a Google folder that has all of these PPTs and different things. So that from me as well as other TAs, all of our stuff is listed there. So I think you can access it. Just go to your portal and you'll be able to find it. Does anyone have any other questions? Because I can send it in the chat, but I think you'll be able to find it very easily there, and I don't have it available with me right now. Everyone else all good? Okay, so if there's no more questions, then we'll all meet again on Monday next week, I think, from 6 to 8 p.m. And you can always send messages. I think there's like a common group where you guys can ask questions, and the TAs and professor can check out your questions and help you through with assignment stuff and all of that. So you can be active there. Um, so yeah, okay then. I guess that's it for today. Thanks everyone for joining. I hope you learned some useful stuff. Yep. All right. Yeah. Great. All right.